Hello guys, we are back to the second lecture, which is lesson 2 of the microbial enumeration, basic calculation and microbial enumeration and microbial growth. And in this lesson 2, we'll be looking into counting method of microbial growth. And in this one, without wasting much of the time, we'll go to the direct method. This direct method is basically the ones we have been having in our practical session in the laboratory, and we'll be looking into direct methods. And the direct method is the most common one and the most prominent one, which all of you have seen in the lab, is the variable plate count. And this is a time honored method of serial dilution and then introducing or inoculating the organism into the petri dish or the broth, so that the microorganism will grow, forming a kind of colony, a aggregate of the microbial cell in the petri dish. You have a colony there, and the colony we can have a valid plate count. The valid plate count ranges between 30 to 300 colonies. Anything below or beyond this is regarded standard by standard process is regarded as invalid. That means anything less than 30 is invalid microbial count or microbial plate count. Anything beyond 300 is regarded as too numerous to count. So we basically do not accept anything range beyond this range. So this is the basic process, and the process always emanates from process called as serial dilution, which we will discuss when we go into lesson 4, where we will now discuss more of the calculation and a lot of mathematical manipulation in order to get and predict exactly, to at least a very precise level, of the number of microorganisms in a particular stock culture. So let's move to the next slide. The second one in the direct method, which all of you will get involved and get experience in the laboratory, is what you call the microscopic cell count using the hemocytometer. So this, which we commonly call a counting chamber, and this counting chamber has series of squares, one big square with a lot of 9 major squares. Inside the 9 major square, you now have another 16 tiny squares inside of them. If you can look at this organism, you can look at this diagram here. You can look at this diagram. These tiny dots are microbial cell. Unlike the first method, which is a culture plate, those ones you count colonies of a microbial cell over 24-hour incubation at a given temperature, which is usually 37 degrees Celsius for those of us in Africa, Nigeria, and other places. You can have other set of organisms that are peculiar to a particular temperature. So it's all temperature dependence, most of the organisms, but most of the organism from which has a normal flora, which has a human being as a normal flora, or environmental, is mostly cultured at 37 degrees Celsius, because that is the normal human temperature at which they survive to enable to cause diseases. So this counting chamber is very important, that we use it when you want to count viable life counts using microscope. When you charge it into this and you cover it with a cover slip, then you view it under the microscope. When you view it under the microscope, this is the image you will see. You will count it and record your values per ml or per volume, which you have charged, usually charged with 1 ml. So let's move to the next slide. The next one is indirect method in the counting system. In this counting system, you are no longer counting the viable cell or the viable colonies, but you are now using the parameters or characteristics of this microbial enumeration growth in order to measure relatively what the growth will have. If you have the first initial growth, you can use the growth rate. You can use some characteristics of the growth. So one is, mostly we usually use the turbidity measurement. In this turbidity measurement, you use a machine or a device called spectrophotometer or spectrophotometry system. In this system is an indirect gauging of the cell density. As the cell density increases within the solution, it tends to absorb light, because this photometer tends to pass light through the medium. Once there is an increase in cell population, the cells will be absorbing the light and allowing fewer light to penetrate. So the spectrophotometer either measure by light absorption, which is what we call light absorption, which we call OD, optical density, or it uses transmittance. That is the amount of light that was allowed to pass through the system. So you can use that measurement over time to gauge either the microbial cell is increasing or decreasing, and invariably it's measuring the turbidity. The second one that is the major type in which we use indirect method is a dry weight measurement. This is where you now have a what you call relative dry mass, biomass of that particular system, and you can be able to estimate the cell biomass using weight measurement. You can use the weighing measurement over time. If you change the change in weight measurement over a period of incubation of a microbial culture, you can gauge the microbial growth in terms of the dry weight. Another one which is not too popular but it's an advanced method of measurement, indirect measurement, is what we call measuring the metabolic activities using the ATP measurement. That's energy utilization or energy inactivity of the microorganism. So if you can be able to measure the ATP level of any system, of any microbial system, you could use that as an indirect method of gauging the microbial activity and as well as the microbial growth. These are the common indirect method we have. However, in the last slide, or which is the completion of this lesson, we'll be looking into modern methods now that are being used for microbial growth and proliferation in any system. The major one of them, which is very common now, is the molecular techniques, where you have your PCR, that is polymerase chain reaction. You have DNA quantification. This has been very, very important in most of the viral infection, just as the last epidemics we have, which is COVID-19 period, which was this molecular system that you were able to gauge the microbial population, that is the viral load within any particular system or within any particular blood sample that was gotten. So these are a modern method. It can be direct or indirect. The modern method cuts across between the two edge. So another one that's also an advanced stage of the photo spectrometer that we have discussed earlier is the flow cytometer or the flow cytometric system. This is a very high throughput approach which measures the characteristics of microbial cells, not directly measuring the cell, but while the molecular method measures the microbial cell or its fragment or its genetic fragments. So that's why we say it cuts across between the direct and indirect, while the flow cytometer, this one measures directly the cells, can be the cells, direct characteristics of the cell, indirect size and shape, or the absorption and the rest of the, just an advanced method of spectrophotometer. Having said that, we are trying to build a preamble to the main calculation which will come up in lesson 4. So thank you as we move to the next lecture.